Chapati, how are you all? So let's just wait for a minute and I welcome you all. Pratima was here, so Pratima, good evening, I was just there, okay. And all my other little champions, I'm waiting for you all, wishing you all a good evening. Let's start this live with a very important chapter, that is molecular basis of inheritance. Yeah? Chalo. Okay, cool. So the rule is, there is one rule in this life, that is everyone who is there has to participate actively, otherwise my motivation goes really down. Okay, Shagufta, hello my dear. Okay, so you have to keep on guessing the answers along with the number. Okay, that question number 1, this is the answer, then only I'll get to know what answer you are saying. Okay, I am great, thank you for asking Shagufta. I hope you are also doing wonderful. Chalo, starting with the first question. The 2 nucleotides of DNA are linked through 3 and 5 carbon of pentose sugar by what bond? N-glycosidic bond, phosphodiester bond, H bond or phosphoester bond. What do you think is the answer for this? They are asking you about the 2 nucleotides. So I hope you know that a nucleotide, okay, a nucleotide is made up of a sugar, and the sugar is bound to a nitrogen base, okay, and also a phosphate group. This whole thing is what you call as nucleotide. Okay, now over here this nucleotide is actually bound to another sugar right there, okay, with the help of a bond like this. What do you call this bond? You call this bond as, any guesses? What do you call this bond as? Where there is a sugar, over here you would be having a third carbon, and over here you would be having a 5th carbon. It looks almost like this. This is the first carbon, 2nd carbon, 3rd carbon, 4th carbon, and there is a 5th carbon having the phosphate group. So this phosphate is attached to the next nucleotide's 3rd carbon right over here. What do you call this bond as? A, B, A, okay, one person says it is phosphodiester bond and the other person says, okay, 1B, actually great, it is known as phosphodiester bond. So how do you name it phosphodiester? Listen, phosphate group is anyways involved, so phosphor should be there. Diester as in 2 ester bonds are there. 2 ester bonds, what do you mean by ester bonds? Whenever something like this is coming together, my dear children, okay, one OH over here and another OH over here is coming together, what happens? H2O is released and you just get one bond right over here. You call this bond as ester bond. So there is 2 ester bonds and in between that you have a phosphate group, which is why you call it phosphoester bond. Okay. Shaukat Ali, okay, Tahsildar, 1B, great. So the answer for this is 1B. Going ahead with the next question that we have over here. Children, if you are not understanding, this is how it looks. Okay, so I can zoom it in and show it to you. This is one sugar, okay, this is whole one nucleotide. Now there is a phosphate group which is going to attach to another sugar on the 3rd carbon. So this is the 3rd carbon and this is the 5th carbon, in between which you have a phosphate group, which is why you call this as a phosphodiester. 2 ester bonds and 1 phosphate group. Cool? Works? Yes, exactly, Pratima. Great. Chalo, let's move on to the next question. In biochemical characterization of transforming principle, which of the following enzymes affect transformation? Which of the enzyme do you think affects transformation? As in, what was in this whole process? In this whole process of transforming principle experiment, it was done by who, children? Who was the person who was behind it? Who did the transforming principle experiment? It was none other than Griffith Bhai Sahab. Okay, Griffith did the experiment where he had a normal strain, okay, where he had a mice who was affected with the normal strain, that is R strain. Okay, now he saw that this particular R strain was transforming into S strain, my dear children, was transforming into S strain, and this S strain is kind of toxic for the plant, sorry, not for the plant, for the mice, that it dies. Okay, because this particular strain is kind of a problematic strain of bacteria. Now, what was used over here such that the transformation was not happening? This R strain to S strain transformation was not happening. Were they using protease, my dear children? Were they using DNase? Were they using amylase, lipase? What was it? So everyone says it is 2B 2B, great. Actually, you're correct. It was DNAs, because the reason why R strain was transforming into S strain was DNA, was because the DNA was kind of secure. Okay, so hence the DNA, when it was passed down, that is when it was getting converted. But now when DNase was used, DNase is an enzyme that will break the DNA off. Okay, that will come and break this DNA off. Hence the transformation would not be happening. So which of the following enzymes affect transformation? It was DNase, not protease. Protease will be affecting proteins, but proteins have no role over here for the conversion, for the transformation. Amylase affects the carbohydrates, again there is no role. Lipase affects or basically breaks down fat, again no effect. So I'll say it is B. Great, Shagufta, Pratima, everyone, Shaukat Ali, everyone, great. Going ahead with the next one. The experiment that proves the semi-conservative principle of DNA replication was given by whom? Who gave the semi-conservative principle experiment? Who showed it? That basically when the DNA is there, when the DNA is replicating and it's making new copy, one strand will be parental and the other strand will be new. Hence, we are basically conserving the parental strand. Half of it is conserved, but it is still conserved. It is not a semi-conservative replication method. Who gave this? Who did the experiment? Anyone? And do children, please do let me know that am I going really fast or am I going really slow, because I don't know, okay, what you actually want from me. So you have to tell me, you have to be vocal. If it's fine, just let me know it's fine. So Shagufta says, Shaukat Ali says, and Pratima says it's D. The answer is D. Okay, so let's see. Is the answer D? Michelson and Stahl experiment, exactly, my dear children. So Michelson and Stahl experiment was done to prove that there is semi-conservative method of replication in the DNA. So you are absolutely correct. And can you tell me on which organism did they do the experiment on? That is also a question that could come, which is why I'm asking you. So D is the answer. Let's move on to the next question. Everyone who got D, great. Hema Varshini, yes dear, I remember you. Definitely. So great to see you over here. Chalo, average rate of polymerization during DNA replication is? Average rate of polymerization, as in how fast do you think that DNA can be replicated? As in how many, the nucleotides can be arranged by the DNA polymerase. So nucleotides have to be arranged by the DNA polymerase. That's how a new DNA strand is being formed, right? What is the speed of this process, whole process? What do you think? Okay, thank you, Shagufta. Thank you for telling me. What do you think is the rate of the process? Okay, Shaukat Ali says it's A. Anyone to challenge? Anyone to challenge? So this question is directly from your NCERT, my dear children. Pratima also says it's A. Everyone pretty sure it's A only? Shagufta says it's C. Yes, my dear children, it's C. It's 2000 base pairs per second. Okay, it's 2000 base pairs per second, not 200 base pairs. Now, this is a thing directly from your NCERT, okay, in the polymerization. So, please make yourself correct. The answer is C. And basically, you're talking about prokaryotes. Eukaryotes generally have a slower rate, but yes. Okay, general question, but this is slightly there in your NCERT, direct question from your NCERT, so we are no one to question it, but it is 2000 base pair per second. So now, questions, my dear children, where am I getting these questions from? Where am I getting these questions from? This is also from the government side only. These questions are additional questions which are given. So you have to practice this. How, as in, this is how the DNA polymerase works. So DNA polymerase has a speed of adding 2000 base pairs per second. That is what it means. The rate of polymerization means. Works? Okay. And this is usually in the case of prokaryotes. So where DNA replication page starts, no, you can just have a read. Under E. coli's replication, they are saying that E. coli does their replication and things like that. Below that, there is a statement where it says that DNA polymerizes with the rate of 2000 base pairs per second. Cool. So I hope this is done. Let's move on to the next question. RNA polymerase 1 transcribes what? I hope, Pratima, you got your answer. Yeah. So RNA polymerase 1 transcribes what? There are 3 different types of RNA polymerase. RNA polymerase 1, 2 and 3. So what does the 1 do? Okay, RNA polymerase, as in it is going to definitely form RNA. And now I hope that all of you know there are different types of RNA, my dear children. Okay, there is rRNA, that is ribosomal RNA, which makes up the ribosome. Okay, and there is tRNA, which will help in translation to happen. There is mRNA, which is the messenger RNA. There is hnRNA, which is heterogeneous nuclear RNA. So you have different types of RNA being made by different type of RNA polymerases. So Kavyashree is there. Okay, someone new out there. Yes, 5B, RNA polymerase 1 transcribes mRNA. Okay, Pratima says 5A. So who is correct? Let's see. Any others who can keep on guessing, my dear children? So over here, I am not going to judge anyone. All of you are my students. So keep on guessing. That is going to make you remember things. The more mistakes you make, probably you will remember better. So Shagufta says 5A. Let's see if it's A or B or whatnot. So RNA polymerase, my dear children, it is going to make rRNA. Okay, there is ribosomal RNA. So I'll say you have to remember these three very important things. Now the question can be RNA polymerase 2 transcribes what? So remember, RNA polymerase is going to make R for ribosome. The first thing that it makes is ribosomal RNA. Okay, rRNA. Now RNA polymerase 2, it is going to do the second work, that is to form mRNA. mRNA is kind of really important, right? mRNA is the one that is going to pass the information. So that is the second priority. So it makes the mRNA. It transcribes the precursor of mRNA, that is heterogeneous nuclear hnRNA. So from DNA, you get hnRNA first, and who does this? This is done by RNA polymerase 2. Further, this hnRNA is modified to form mRNA. So RNA polymerase 2 does this. RNA polymerase 3 does what, my dear children? It helps in the formation of tRNA, 5sRNA and snRNA. That is totally different. But you should be knowing all of this. Is this clear? If it's clear, give me a thumbs up. Everyone who is out there. If you think that this particular portion is clear, and if you get any question from this particular portion, you are going to score, give me a thumbs up over there in the live chat. Then only I will be understanding, right? Otherwise, I'd be feeling that basically I am going very fast, or I don't know. Okay, no one says that you have understood. So no one has given me thumbs up, which means no one understood the previous part. My dear children, please, it's not always the same question would be repeated. Do not think like that. The questions would be reframed. The pattern would be the same. Yes, Shagufta understood. Great. Thank you so much for considering that. Pratima also. Shaukat Ali also. Great, my dear children. Chalo. GGU, GGC, GGA and GGG all code for glycine. So glycine is an amino acid which is coded by all of these codons. Now which salient feature of the genetic code does it prove? Does it show? What do you think? Which feature? You know, genetic code has different features, that it is universal, it is unambiguous, it is degenerate, and this and that, lot of things are there. Which one you think is the correct option? So it is saying there is one amino acid, that is glycine, and it is coded by X, it is also coded by Y, it is also coded by Z, it is also coded by C. So many codons are coding this. What feature does it show? Shagufta says 6B. Degenerate codons. Okay. Shaukat Ali says 6C. Okay. Universal code. How can it be universal code? Universal code is when basically we can say, for example, I have the codons, this codon for glycine, and E. coli also have the same codon for glycine. That is when I say that the codon is universally same for everyone. So I cannot use the universal codon over here. And unambiguous as in it is not basically, it does not have comma, full stop and things like that. Yes, my dear child, please ask the doubts. So basically over here, when you say there are so many codons coding for one particular amino acid, you call these codons as degenerate codons. Okay, you call them as degenerate codons. You can remember like this. You think that you have a best friend, but your best friend has so many best friends. So it is a very sad thing, right? It is a degenerating thing. That's how I remember it, okay? So many codons for one particular amino acid, you call this degenerate codons. Okay, even though you should not be doing anything to your best friend, he or she will be really good person. Okay, chalo, I hope this is clear. So your answer is 6B. Okay. Nayan, please ask me the doubt, my dear child. So some amino acids are coded by more than one codon, hence the code is degenerate. That is the answer for this. Cool. Let's go ahead. Aminoacylation of tRNA occurs during when do you think aminoacylation of tRNA occurs? What do you mean by aminoacylation of tRNA? So the tRNA guys, no, it looks like somewhat like this. So it's normal RNA, but it is twisted. Okay, so the 5' end is over here and the 3' end is over here, and this codon, this loop that you see, you call it as anti-codon loop. So what it does is that this anti-codon loop goes and binds with the codon of the mRNA. Okay, it goes and binds with the codon of the mRNA. Now, what it is coding? What is this tRNA actually doing over here? It has to carry amino acids. It has to carry amino acids. So each amino acid has to come and make a bond over here in the 3' end. So this process is known as aminoacylation of tRNA, or what do you call it? You call it charging of tRNA, either way. And when is it important, my dear children? When you want the information right over here, that is the information in the mRNA, to be translated to form a protein. I don't understand this information, so I need help of a friend, that is tRNA, to make myself understand this and to make a protein out of this. What do you call this process? This process is known as translation. So B is the answer. Who all got it correct? Let's see. Shagufta got it correct, Shaukat Ali got it correct, Pratima got it correct. Great. Okay, wait. Can we complete studying for boards in 1 month if we completed syllabus half and half? Yes, you can. Nayan, you can definitely do it, my dear children, dear child. I am so happy at least you have completed half and half, so it's okay. Keep going every single day. You have to study. Do not have to lose even a single day of preparation. You will be able to ace it. Okay, that's my guarantee, but 100% should be your guarantee. Fine. So Pratima, everyone doing really great. I'm going ahead with the next question. So aminoacylation, yes. Now this is a question which I think, which is why I put it in red. Okay, you need to use a little of your brain. This is something that I loved so much. So in a single stranded DNA, if the ratio of adenine and guanine is 60 and 40, then the ratio of thymine and cytosine is what? Can you tell me? So it is a single stranded DNA and the ratio of adenine and guanine is what? 60 and 40. You are asked about the ratio of thymine and cytosine. This is a very tricky question and a very nice question. So usually what we do, so adenine is 16, so we assume that, okay, if in one strand, if there is adenine and if there are 60 of them, obviously adenine binds with thymine, so thymine will also be 60, right? That is what we think. And if guanine is basically 40, then obviously the guy it pairs with the cytosine, it will also have 40. So we think the answer will be what? Basically it will be 60 by 40. That is a usual thing. But do you think it is correct? Again, someone did that mistake. Okay, Shaukat Ali, Shagufta, everyone, it's okay to do the mistake, no problem at all. I love it when you try your level best. That is the best part of the live sessions. So everyone keep on guessing. Shagufta says 8B, fine. Let's see, let's consider that. Can I tell the answer? Say yes or no. Shall I tell the answer, or do you want me to wait for 50 seconds? Fast. I am giving you a chance to do this question. Read each and every part of this question carefully. You will be able to do it. Okay. Pratima, lock. So she's saying, okay, let's lock 8B. Yes, chalo. I'll say, my dear child, it is basically single-stranded DNA. So can I consider this part of the DNA? So over here, if I am saying that adenine is 60, guanine is 40, again, thymine can be any number. Cytosine also can be any number. It's about the single-stranded DNA. I'm not asking about the base pair thing. Okay, it's not a double-stranded DNA. So answer will be what? None of the above, because we do not know. How can we know? In a single strand, if the ratio is the ratio of this, then the ratio of T and C is none of the above. We did not know. How is the question? Is it lit? If it is lit, give me the fire sign or tell me it's lit. So I love this question because of this particular reason. I hope you understand. There is no double-stranded DNA. There is only single-stranded DNA. If A and G is this, there can be many amount of T also. There can be much amount of C also. You get it? Will you be able to answer this question on in the boards if it comes? So the answer is D, none of the above. How did you like the question? Was it good? So please do not be scared for this. It's okay. When I read the question, I'll tell you honestly, even I did the same thing just like how you did. Then I was like, there is some catch. How can they give such easy question? So yes, great Shagufta. Thank you for acknowledging that is a lit question. Okay, chalo. Now question number 9th, my dear children. The components of deoxyguanosine is. The components of deoxyguanosine. What do you mean by deoxyguanosine? That means there is a deoxyribose sugar, and on the first carbon of this deoxyribose sugar, what do you have? You have guanine. Guanine is a nitrogen base. Now, when guanine comes along with the sugar, no, that is deoxyribose sugar, you will now call this thing as deoxyguanosine. Okay, what do you call it? Deoxyguanosine. So, what do I need for it? My dear children, I need guanine and also deoxyribose sugar. So, in DNA, you have deoxyribose sugar. In RNA, you have ribose sugar. In RNA, there is an OH group up over here. In DNA, we do not have this OH group up over here. Cool? So, which is making it deoxy. There is O is missing. So, deoxyribose. Works? Chalo. So, 9 is C. 9 is C, yeah. How can 9 be C? Guanine, deoxyribose and phosphate. No, Baba. Phosphate would not be there. If you are adding phosphate, what will I call this? If I am adding phosphate, I will call this deoxyguanosine monophosphate. I will call this deoxyguanosine monophosphate, as in just have one phosphate. Okay, so the answer is B. Cool? Chalo. Enjoying the class? Children, we are far with 10 questions. We have 20 more questions, 30 questions in total. Are you enjoying the class or is it boring? So don't forget to hit the like button. Okay? Chalo. I am also enjoying the class with you guys. So the coding sequences present in processed RNA is called. What do you mean by processed RNA? As in the one which have got completely modified and things like that. Okay, so the coding sequences, the sequences that code. If you do not know, let me tell you, if this is a DNA, my dear children, okay, if this is a DNA, there will be areas which do not code for anything, proteins, and there will be area which will be coding for something. Okay, so the things that code for something, you call them as exons, and the things which are intervening in between these exons, what do you call them? Introns, my dear children. They are the ones that do not code for anything. Now, when mRNA is made after the DNA, it is made sure that, okay, first thing I told you, the DNA which is there, it transcribes into hnRNA, heterogeneous nuclear RNA. In them also you have introns and exons and everything. Then this hnRNA undergoes something which is known as processing. It undergoes processing and says that I do not want introns, I just want exons altogether, I just want the coding sequences. So all the exons, they line up like this, and now it's just pakka coding sequence, and this is what you call as mRNA. So the coding sequence is present in processed RNAs. This is the processed RNA. What do you call this? Yes, non-coding is introns. Maths and maths is here. So I am seeing for the first time someone with maths and maths name coming and helping in biology. Great. Kudos. So Shagufta, Pratima, Shakat Ali, every one of you, maths and maths, Pratima, every one of you is correct. Great. Kudos to you, my dear children. So answer is B. Fine. Now 11th question, again something that you have to use your brain, lit question. So, if a processed mRNA of a particular cistron contains 636 base pairs, so there is an mRNA and this mRNA consists of 636 base pairs, that is, okay, base pairs, you know that codes for polypeptide chain, as in this mRNA is going to make what? Proteins. So, this chain of, let's say, small amino acid, you call them polypeptides. Now, what are the number of amino acids that it codes for? What are the number of amino acids that it codes for? So, I'll tell you one basic math, okay? 3 codons come together to form one amino acid, to code for one amino acid. If you have 636 base pairs, in the 636 base pairs, my dear children, all of them basically have to come in the group of 3333 codons and give rise to how many amino acids? That is the question. So if 3 codons are coding, that is 3 base pairs, okay? If you're confused about that, okay, I wouldn't say 3 codons, that will be wrong. So sorry, so sorry, so sorry. It is 3 base pairs. Okay, 3 base pairs comes together to form a codon, and this particular codon codes for 1 amino acid. And if you have 636 base pairs, how many amino acids will be formed? Can you tell me? 212. Okay. Shagufta and Chakatali says 11A is the answer. Does everyone think the same? Pratima also says it is 11A. So shall I lock it? Shall I lock it? Chalo. I'm locking it. And I'm going to tell you the answer. It's not 11A. The answer is 11B. Why? Because I told 636 is there, base pairs is there in the mRNA. Now the last one which is there, obviously if you have to make a polypeptide chain, that there should be some stop codon, my dear children. There should be one stop codon right there, which not be coding for anything. Which would not be coding for anything, is just there to stop the process of translation to happen. So your 3 base pairs are waste. Your 3 base pairs or basically codons are waste. So if I calculate it like this, you calculate it like this, right? So if 636 divided by 3 is equals to 212. If I am removing one set of codon, if I am removing one set of amino acid, what do I get? The answer will be 211. Okay, if you still didn't get, I'll tell you. 636 is the total number of base pairs. You're going to make them into 3 groups. I get 212. Okay, so each 3 base pairs over here are going to code for 212 amino acids. But you forgot about the fact, okay, you forgot about the fact that the last codon which is there, it is a stop codon which does not code for any amino acid. So I have to forget about that. So the answer will become what? 211. So I have to remove one amino acid from there. It will be 211. Hence your answer becomes B. How was this question? Tell me. Come on people, are you listening to me? Why don't I see a reaction? At least being a teacher right over here, I'm talking to you and giving myself entertainment by talking myself. But you should also be talking to me. Where in the comment section over here, right in the live chat, can you tell me? Okay, maths and maths says clearly understood, ma'am. How was the question? Is it lit? Tell me if it's lit or not. So I know most of you might be thinking, oh, what happens if this type of question comes my? It's okay. This type of questions have to come. It will be coming like that, because if they give you easy questions, everyone will be scoring really good, and there are a lot of things ahead in the education system which won't allow you to do so. So you have to do your preparation 100%, you will be able to do this. And what's the basic thing that you have to remember? That is, you have to read the question properly. You will get the marks. Okay? So this is the answer. And I am really proud of you guys. You will be doing it, I'm pretty sure. Now question number 12, maths everyone, thank you guys. Now if a mRNA, that is 3-AUG-UAA undergoes translation, how many types of amino acids are present over here? Again, it is tricky. I have not made the color different, but it's tricky. Read the question properly. That is what I mean whenever I say tricky. It's not that the question is difficult. So you have to see how many amino acids you can make out of it. That is the thing. 3-AUG and this is 5-. I'm telling you, each codon which is there, made up of 3 base pairs, X, Y, Z, is going to code for 1 amino acid. And you are not asked how many number of amino acids are being formed. You are asked how many types of amino acids. Okay, how many types? So basically, I'll give you a clue. If an amino acid is being repeated, you don't count it. You just count it once. Chalo bachcha log, keep on guessing. Tell me. Okay, I'll give you some shanti to think about it. From when the time I start saying tick tick 1 to tick tick 5, you have to tell me the answer, okay? Tick tick 1, tick tick 2, tick tick 3, tick tick 4 and tick tick 5. Shagufta says it's 12B. Okay, what about others? Shaukat Ali, Pratima, Maths and Math, and everyone who is there in the live session, why are you not coming in front and talking to me? I won't be doing anything. I'm a very cool teacher. Trust me on this. Okay, anyways. So Shagufta said it is 3 type. Let me trust Shagufta over here and let me try to decode the answer. Okay, so my dear children, always remember mRNA is read from the opposite end. If you are reading from this end, you will realize, okay, you will see, oh, UAA is there, UAA is a stop codon, so it won't be making any amino acid over there. It will be wrong. Always remember, in 5' to 3' end, you have to keep the mRNA. Right, if you are keeping in that sense, how do you read it? From here, you have to read it, AAU. So that is a codon coding for one amino acid. Now, UUU is a codon coding for another amino acid, and then again you have what, my dear children? Wait, just a second, okay? UUU again coding for the same amino acid, so I won't be adding it. UUU again coding for the same amino acid, that is phenylalanine. Now you have what? GUA. GUA is the 3rd type. So how many type of amino acid is there, my dear children? It will be 3 type. It will be 3 type. Always remember, do not do the mistake of reading it that side. Why? Because the 5' end is over here. You have to read from the 5' end. So AAU is 1, UUU is phenylalanine, again UU, phenylalanine, phenylalanine, and then you have GUA. So it is 3 types of amino acids that are being formed. So what is the answer for this? It becomes B. Cool? Yeah, you're talking about the stop codons, right? UAA, UAG, UGA is actually a stop codon. So this is just kept to confuse you at the end. But you should be knowing you are reading from this end, so it won't be the stop codon. It is read as AAU. Cool? Chalo, great. Great Shagufta and great Pratima for telling that's a stop codon. Chalo, drawing really good. Now, 13th question. Statement 1. Deoxynucleotide triphosphate dNTPs acts as substrates during DNA replication, as in the nucleotide triphosphate which is there, okay, that is this sugar thing which is there with the nitrogen base, and actually there are 3 phosphates attached, but when we want to make the nucleotides arrange together, we break this bond and we just have 1 phosphate. That is how it works. So you call this what? You call this as deoxyribose nucleotide triphosphate and 3 phosphates. So it acts as a substrate during DNA replication, as in it means that you are basically taking their help to make the DNA replicate, or for the DNA replication process this is used. This statement is correct. What about statement 2? Deoxynucleotide triphosphate also acts as a source of energy in the process of DNA replication. Choose the correct statement. Do you think the second statement is also correct? It says that this particular thing that you see, it is also going to act like a source of energy for DNA replication. Do you think it is correct? Tell me in the comment section. Come on, where are you all? Pratima says 13A. So you mean that both the statements are correct. What about others? Bacha party, if you are not telling me the answer, I also won't tell the answer. I am cutting. Come on, children. Okay, Shagufta says it's 13A. Okay, so everyone is coding for AAA. Can you explain why? Why? Any guesses? I'll tell you for the people who do not know. It's okay to not know, okay? So I told you there is triphosphate. And I'll tell you one thing. Whenever you are making the DNA molecules, I told you this bond is being broken. When this bond is being broken, my dear children, there is lots of energy being released. There is lots of energy being released over here, stored in these bonds, such that it helps in the formation of what? It helps in the formation of bonds between these nucleotides. It helps in the formation of DNA. So because this bond is broken, whatever energy is being stored over here is being utilized for formation of another bond. You get it? So which is why I'll say both statement 1 and the statement 2 are correct. This is correct. There's a new person over here. Shweta R says 13A. Great. So entry is good. And Math and Shagufta. Okay, it's not C. Can you tell me why it's C? Why do you think it's C? It's not C, but it's okay. You tried. And now since you know, I hope you will be doing the correct answer in your board examination. Okay? So it will be both the statements are correct. Chalo. Children, I hope you're understanding, yeah? So in pea plants, each character is governed by a gene which is monocistronic, polycistronic, operon, intron, whatever. What do you think? In pea plants, each character is governed by a gene which is monocistronic, polycistronic, operon, or what? What do you mean by monocistronic? Monocistronic is when basically what happens is that you have a gene, and for this gene you have a separate regulator and promoter, operator, everything. Okay, there's a separate thing over here for one particular gene. This is what you mean by monocistronic. Okay, and this is found in eukaryotes. Always remember, monocistronic genes are found in eukaryotes. What about polycistronic? Where what happens? For example, suppose this is the DNA, okay, or whatever, mRNA. Over here you have gene 1, you have gene 2, and you have gene 3. For all of them together, you will be having a common promoter and common. So over here you see multiple genes are there. You call them as polycistronic. Polycistronic, and this is found in prokaryotes. Why am I explaining you this? Because this question can come. So in the case of pea plants, plant is what? Eukaryote. Okay, plant is like us. We all are eukaryotes, my dear children. So answer will be what? A. Sampreet says not visible. Is it the same for everyone else? It is visible, my dear child. Okay, I am also seeing my live. It's fine. Can you please reload it once? And okay, 14th answer. Only Shagufta has said the answer, and she says it's A. What about the rest of the children? What about you guys? Okay, sorry. I'm so sorry. I'm so sorry. I'm so sorry. Forgive me. So it is Ayo. It is polycistronic. Okay? And polycistronic thing is found in prokaryotes. I'm so sorry for the mistake. I hope I have rectified. And everyone else is 14A. Pratima and Hafima. Wow, new person. Nice name as well. So Nishmita, I hope that query solved. And Trabhufta, Pratima, Halima, everyone. Great going. So I see the girl crowd. What about boys? Where are the boys? Girl power is going really strong over here. Great. Chalo. Now which of these is correct? So Mamta also said B. Baba, B is not correct. It is not polycistronic. I told you polycistronic is found in what? In prokaryotes. Prokaryotes as in bacteria and things like that, like E. coli and them. Okay? Chalo. Next question. 15. Which of these are correct? Okay. UTR, untranslated region, are located on the DNA. Second, UTR is present both at 5' before the start codon and also at 3' after the stop codon. UTR provides stability to mRNA. UTR is required for efficient translation. Can you tell me the answer? Please answer like this, okay? 15, A, B, C, D, whatever you want to say, you can say. So what do you mean by UTR? UTR is an untranslated region. So if this is an mRNA, my dear children, okay, mRNA, there will be start codon, that is AUG. There will be stop codon as well. For example, UGA or things like that, whatever. This is the start codon and this is the stop codon. So in front of this, there are certain regions which are not translated. You call these regions as untranslated regions, and they are really, really important. For what? For basically the process of translation to begin, for the process of translation to get initiated, for the mRNA to be stable, everything, every single thing they are responsible for. Okay, so in this statement, okay, so I see answers coming around floating over here. Nishmita says it is A. Okay, okay, great. Shagufta says it's A, and again, great, great. That's how you should be answering. 15th A. So over here, this is the, which of this is, sorry. Which of this is correct? We have to say UTR, untranslated regions, are located on DNA. No, children. It is located on mRNA. So I'll say this is wrong. Okay. Now second is UTR is present both at 5' at the start codon and also at the 3' after the stop codon. That is correct. UTR provides stability. That is correct. UTR is required for efficient translation. That is correct. So 2, 3 and 4. 2, 3 and 4. How it is 15A? How is it 15A? Tell me. A means 1, 2 and 4 is correct. But I told you, untranslated regions which are there, they are going to help with the initiation of translation. They are going to make the mRNA stable. They are present at both the end. So how is it correct? How is 1 correct? And this is mRNA. Here what is present? DNA. It's not there. Okay, it's not there. Understand? So the answer, everyone tell me the correct answer now. 15th D is the correct answer. Yes, 15th D is the correct answer. I hope everyone gets it. Mamta Baba, you're able to understand? Or do you want me to explain? Yes, yes Shaukat Ali, yes Halima, yes Shagufta, Pratima, everyone. Great. Mamta Baba, I hope you understood. Okay? Chalo. Going ahead. During transcription, the site of DNA molecule at which DNA polymerase, this is not DNA polymerase, let's make a quick change, RNA polymerase, okay. During transcription, the site of DNA molecule at which RNA polymerase bind is called, okay, Mamta, forgiven my child. Can you tell me the answer? A, B, C, D. When transcription is happening, no, of the DNA molecule, at which site the RNA polymerase binds? What do you call this? 16A. Yes, it is the promoter sequence. This is a direct question. Okay, very direct question. So I hope you know there is a promoter region. Okay, and in between this you have the structural gene and there is a terminator region. So promoter region is where the RNA polymerase will come and bind and start transcribing this particular DNA to what? mRNA. So this is the promoter region. So Shaukat Ali, Nishmita, Shweta, Pratima, Shagufta, Halima, all of you. Great. Great going champions. So it is A. Easy question, no? Now tell me this question. 17th question. Methyl guanosine triphosphate is added at the 5' end of hnRNA in a process of. What is hnRNA? I told you, when you say DNA transcribes to form mRNA, does it actually form mRNA immediately? Nahi. That's a scam. Actually it forms what? hnRNA, heterogeneous nuclear RNA, which has the introns and the exons and everything. Now this guy undergoes a lot of processing to form mRNA. And while it is undergoing processing, as I said, splicing is the first thing that happens, where introns and everything are removed and you just have exons, the coding sequences all together. Then something is happening that is known as capping. What do you mean by capping? Capping is where at this position, at the 5' end of the hnRNA, okay, what are you going to do? You are going to add a group that is methyl guanosine triphosphate. Okay, you are going to add methyl guanosine triphosphate. Why do you do so? Always whenever someone asks you something, okay, whenever you're asked a question, you should be knowing the why, how and everything. Okay, it's not about marks, it's also about knowledge. So why is it done so? Because this mRNA guy, no, okay, this mRNA guy has to make proteins, and now proteins will be made outside the nucleus. So when this mRNA is going to sneak out of the nucleus, what happens? There will be so many enzymes that could act on it. So to save it from all the enzymatic activity, you have methyl guanosine triphosphate, and also to show it the direction that yeah, you have to make a protein. That is also why you have capping being done. Okay, now tailing is where you are adding adenosine, AAA, so many things. That is totally different. So the answer is capping, as you all said. Who all said? Let me see. Okay, it is the 17th question, right? So Shaukat Ali, 17B, Halima, Baba, I hope you got the answer now. It is capping, okay, it's B. Shagufta says 17B, great. Mamta says B, Nishmita B, Pratima B, Shweta B, great, 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 great. Great guys. So Shaukat Ali is the only guy right here who is answering everything. The guy gang who is there, you guys also should show up. Okay, great. Children, did you have water? Did you hydrate yourself? If not, drink water. Okay, now question number 18th. If the template of DNA strand is this, the template of DNA strand is 3 dash its complementary DNA strand would be. What would be its complementary DNA strand? It's a very easy question. Everyone can do this. Okay, so this is, I am writing just like this, A, T, G. Is this visible? I hope it is. Okay, A, T, G, C, A, T, C, T, and then you have what? 5' end. This is the 3' end. Okay, now you have to make a complementary strand of it. What is a complementary strand? 18th, Baba, C, A, everything is coming, huh? C, okay. Okay. Let me see now. Okay, so when you are making a complementary DNA strand, A always binds with T. This is 5', okay. T with A, G with C, C with G, A with T, A with T, T with A, C with G, and T with A again. This will be 3'. This is the thing that you have to find. So T, A, C, G, T, T, A, G, A. You all are correct. Ma'am, this question asked, okay, this question was asked in preparatory exam. I told you. From this, this is given by the government itself, which is why I am taking on priority and making you solve these questions. Okay, so I also saw a question came in Bangalore North Zone question paper, which was from this. So please make sure you're practicing all the questions really well. So this is C, my dear children. Chalo. Now easy questions I would be able to see, at least a few of them. Okay, so expand YAC. What is YAC? Can you expand YAC? By that time I will just have a glass of water and come. Oh sorry, not glass, a cup, not a cup also. What do you call this? A bottle of water. I have little water left. What do you mean by YAC, my dear children? So I want to tell something. I'm really happy that a lot of you came over here. I was saying, Aishwarya ma'am, for biology there is no crowd. So I was kind of sad because of it. It's 19th question. Can you tell 19th? I'm in 19th question. 19th B. Expand YAC, East artificial chromosome. Yes, it is East artificial chromosome, my dear children. So this is a plasmid. Okay, you know plasmid, they are vectors. What is plasmid? A plasmid is a self-replicating unit of DNA. Okay, but what do? This particular thing, I am going to cut this portion and I am going to put the gene of interest that I want. Suppose I put my gene of interest over here. Now what happens? This particular DNA strand will keep on replicating and things like that. So basically my gene of interest is also replicating. So if I want my gene to keep on replicating, to give me more and more products, I will basically make something like this, which is known as East artificial chromosome. Nishmita Shetty, it is BAC and I think when I am not able to recollect, it is towards your, before your DNA fingerprinting and things like that. When vector is being talked, when vector is being said, at that point you have YAC and BAC. Towards the end, the DNA fingerprinting part, you have this. Can you just check? So Pratima, B, oh my god. Shagufta B, Shaukat Ali B, great. Mamta, D. Abba. It's not D, it's not East artificial chromosome, it will be East artificial chromosome. Artificially you are making it. Okay? Chalo. Next question. Expand BAC. Next question is also kind of same. BAC. Easy. You're most welcome, dear. BAC is what? 20th answer. Now we have 10 more questions and then we are done, okay? You will be seeing some good questions later on. Can I tell, is the live really slow? Why am I getting slow reply? Are you waiting for me to answer? No, you had to answer by yourself. 20 is B. 20 is B. Okay. So someone said 20 is A. Pratima said it's A. Let me see. Bacterial artificial centromere. Abba. No. Bacterial artificial chromosome. This is the answer. Okay, just like how you have East artificial chromosome, you have bacterial artificial chromosome. Cool? So it is 20. Shaukat Ali and Shagufta, you're absolutely right. It's B. Chalo, this is the question that I said that I saw in a question paper that I did yesterday in the preparatory exam video. If you have not seen, I have got the same question being repeated. Shweta, 19th BR. Yeah, I'm an engineering student. I didn't, okay, maybe because of your voice. Oh, so thank you. I hope you study a little of biology as well with me. Okay, chalo. Match the following, my dear children. The easiest way I'll tell you, okay, to crack this, if you do not know all the names. I hope everyone knows Griffith. Griffith did the transforming experiment with the mice and all. Okay, so Griffith, D goes with transforming principle. So D goes with 2. Let's find D with 2. So there's only one option who has D with 2, and that is this option. Rest of all the options I can cut off, but let's see other answers also. Now I also know one more thing. Jacob and Monad are the people who gave the lac operon. Jacob and Monad are the people who gave lac operon. Yes. Yes, yes, C is with lac operon, so C is with 1. So that is also something I know. Now if I see the rest, it's easy. Alec Jeffrey, who is there, he goes with 4th. DNA fingerprinting was introduced by Alec Jeffrey, and F. Sanger, Frederick Sanger was the person who annotated DNA sequencing. So this is the answer. Okay, so what is the answer? 21, the answer is B. All cool. Nishmita, Shagufta, Pratima, Mamta, Shaukat Ali, great bacha party, superb. Okay, chalo, let's go to the next question. Okay, if A and B are the DNA of 2 mothers and C is the DNA of the child. So A and B, if you can see, is the DNA of what? 2 mothers, and this is the DNA of a child. They have not mentioned whose child and things like that. On observation of banding pattern of DNA fingerprinting, it can be concluded that, what can be concluded? That child C belongs to mother A, that child C belongs to mother B, or A and B both are sisters, or no conclusions can be drawn. What do you think? Basically, always remember, we all have something common in between us and our parents. Okay, that is some repeated sequences. So in our DNA also we have those repeated sequences, and in their DNA also there is repeated sequences. Now there might be changes also, which is why me and my parent are totally different from each other. There will be changes also, but the repeated sequences, if we see, they are almost common. So you have to spot the DNA which is kind of having the same type of bands. Okay, that means they have some similarity between them. So 22 is B. Shagufta and Shaukat Ali and Pratima says it's B. Is it actually B? Let's see. So child C belongs to mother B. That is what B says. And kudos to you. The answer is absolutely correct. Over here, see the pattern really carefully, okay? Zoomed in, enough zoomed in, okay? So you see red pattern over here, red band over here, red band over here, red band over here, yellow, 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 blue, 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 blue. Everything is kind of same. Even though some changes might be there, but it's not been shown up here, but it's almost same. Okay, so child C is actually belonging to mother B, and this DNA fingerprinting method is used when there is paternal maternal issue. You do not know who is the parent of the child. That's when this particular thing is greatly being used, and also during criminal cases. So I see a new person over here, T.R. Gaber. Great Baba, you said 2 answers. What should I lock? It's B. The last one you said, I'll lock it, okay? Great. Chalo, 23rd question. 23rd question we'll do. In biochemical analysis of transforming principle, only DNAs could not transform R strain to S strain. R strain to S strain. So, I told you a mice was having a strain that is rough strain. But later when, not the mice was having the strain R strain. Basically, when R strain was mixed with S strain, some problem was happening. Okay, some problem was happening such that this particular R strain which is there, it was also getting converted to something that is not really good for the mice. It was killing the mice. And we were trying to find out what is this transforming thing, what is this principle which is helping in the transformation of R strain, which is usually Papa guy. Okay, it is converting it into something virulent. Who is that? So it was later found that it is DNA which was actually transforming this R strain into a toxic form, that is the S strain. Okay, now if I use DNAs, that is enzymes that basically degrade DNA, obviously this DNA would not be there, and hence transformation would not be taking place. Okay, DNase will attack this, and obviously this transformation is not going to take place. So in biochemical analysis of transforming principle, only DNase could not transform R strain to S strain. Yes, that is true. If DNase was there, then there is no transformation also. So statement A is correct. Okay, you have already told me the answer. Pratima, Shaukat Ali, everyone is saying A. Let's see. And someone says D. Let me see. So statement 2 says, this experiment proved that proteins and RNA are not the transforming principle. Did this experiment prove that proteins and RNA are not the transforming principle? Yes, it definitely proved that. Why? Why? Because when I treated with protease, since transformation was happening, when I treated with let's say RNAs, then also transformation was happening. That means even if proteins are degraded, even if RNA is degraded, tabhi what say transformation is definitely going to happen. But if DNase is used, transformation is not happening. So I got to know this from the experiment being done. So I'll say statement 1 and statement 2 both are correct. So A is the correct answer. So who all got A is absolutely correct. So Shagufta and Shaukat Ali and TR Gamer, Pratima, Nishmita, everyone is reading. So that you're reading. Okay, everyone is absolutely correct. I think yes, there is some issues because I am seeing your comments really late. Okay, it is A. Now BA Creation says, I am attending this class because I want to teach my little sister. I am encouraging her to choose medical field. I hope your classes make me a biology teacher. Definitely. I am so happy to see that comment. Okay, I hope your little sister also becomes something that she really wants, and you also become something really good that you want, whatever it be. Okay, even if it's not medical field or if it's medical field, whatever. Chalo. Great. Chalo, thank you children for and I think Shaukat Ali, if is it fine now, the pace is fine, or are you facing the same thing? Anyways, let's go ahead with the next question. Okay, you tell me in the comment section, I'll be seeing it. What are the structures called that give an appearance as beads on string in the chromosome when viewed under the electron microscope? So the chromosomes, they appear to be something like a bead on a string like structure. So what is this thing? What is the reason why they appear as bead on a string like structure? Shaukat Ali says 24C, straight. Okay. TR Gamer also 24C. Shagufta says B. I think you're confused a bit. Pratima also says B, but let me tell you the answer. The answer is what TR Gamer and Shaukat Ali said, it is C, also Shweta. Okay, so my dear children, what happens? The DNA is pretty long, okay, and our nucleus is kind of small. So you have to arrange them in a compact manner. So what do? Basically, you have the chromatin fibers first. There is chromatin fibers, that is just the DNA, okay. These chromatin fibers, they arrange around a structure which is known as nucleosome. How many rounds does this chromatin material do? They take 2 rounds. So almost 200 base pairs of DNA is making a round over here in the nucleosome. In this way, what is happening? You are basically making a compact structure over there. You are basically making a compact structure. So the DNA along with the nucleosome that you see over here, it is going to give a bead on a string like appearance. Okay, this particular DNA is again going to go again, it is going to wrap around another nucleosome. So it is nucleosome. Okay, great. Mamta, Nishmita, Pratima, everyone. Sorry, it's okay. Children, can you tell me one thing that is not there in the question? What is the nucleosome made up of? What is the nucleosome made up of? Which protein? Can you tell me that also? Because that is also important. And how many proteins come together to form a nucleosome? 2 questions. Nucleosome appear as repeating units on the chromatin in the nucleus. Great, yes. True. There are 2 questions. What proteins makes up the nucleosome and how many proteins have to come together to form the nucleosome? Nishmita says histone is the protein's name. Exactly, histone is the protein's name. And how many of them do you need? How many of them do you need? Histone protein, Shaukat Ali also. 8 unit of histone. Great, yes, 8. So 1, 2, 3, 4, and then backside also you have 1, 2, 3, 4. Okay, 8 of them will be coming together, and the whole thing that you see over here will be known as nucleosome, my dear children. Okay, nucleosome is said when you have all the histone proteins. What do you call this? You call this as octamer of histone. octamer as in 8 histone proteins are coming together over here, and the DNA guy is just going to chumma wrap around over here twice. Why? Because DNA is negatively charged. Remember, DNA is negatively charged, and the histone proteins which are made up of amino acids, they have positively charged amino acids over there. So positive and negative comes together, which is why it is able to wrap. And now this structure, you call this as chromatin. When everything comes together, all the nucleosomes comes together, you call them chromatin. The chromatin, when it condenses together very close, what do you call this structure as? You call it as chromosome. So DNA is there, DNA wraps around histone and then makes a string-like structure. You call the whole thing as chromatin. Chromatin condenses to form the chromosome. Okay, done? Histones has arginine and lysine. Yes, it is not protein. Histone is a protein. Okay, histone itself is a protein, and proteins are made up of what? Amino acids. Now, there are 2 amino acids which are richly found in this protein, and that is what you said, arginine and lysine, my dear children. These both are positively charged, the DNA is negatively charged, so it laps around. So good. Shagufta, really great. Histone proteins and 200 base pairs. Yes, 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 yes. BA creations, everyone. Oh my God, I am so fascinated by seeing you answer. Okay, good going. Chalo, let's go to the next question. In a DNA strand, if there are 100 nucleotides and 30 are adenine, the number of thymine, guanine, cytosine respectively are. So, in the DNA strand, my dear children, okay, 100 nucleotides are there. And okay, 30 is adenine. 30 of them is adenine. How many number of thymine, guanine, cytosine do you have respectively? Can you tell? (25A). 25A. Shaukat Ali said 25A. Let's see. My dear children, all the adenines are going to form pair with thymine. All the guanine are going to pair with cytosine, right? So, adenine's number we know, that is 30. So, obviously thymine will also be 30. Agreed? Now, these are 30, 30. These are 30. Total, how many nucleotides do you have? You have 100. So, 30 and 30, 60 is done. 60 is already done. Now, you are left with what? 40. You are left with 40 more nucleotides. Okay, so if I say guanine is 20 and this guy is also 20, can I say 40, 40? Sorry, it has made 40. So, 30 plus 30, 60 plus 20, 80, 80 plus 20, 100. So, the number of thymine will be 30, the number of guanine will be 20, and the same will be the number of cytosine, 20. So, what is your answer? 30, 20, 20. A is the answer. And Shaukat Ali was the first one to answer this. BA creations, great going. I see you are also interested in biology. That it's not just for your little sister, I'm pretty sure. You also know a lot of biology. Nishmita 25A, great. Shagufta 25A, Pratima 25A, Mamta Baba 25A. Everyone is correct. Kudos to you. Chalo. Now last 5 questions. Let's do it. I hope for everyone it's clear. Always see the calculation and see the number. What is the mistake that you could probably do? I'll tell you that. So you see 30 is there. Now you minus 100 minus 30. You do 70 and then you try to find too much difficulty and then you say 35, 35. No, it's not like that. Okay. So please just be careful for that. Chalo. Next question. The chemical method developed by Hargobind Khurana. So that is a boring question, but also we need a boring question in between, right? So the chemical method developed by Hargobind Khurana, who was a scientist, my dear children, was, what was his chemical method? Synthesizing RNA molecules with defined combination of bases, synthesizing RNA without a template, categorization of RNA, cell-free protein synthesis. Now this is a question directly from your NCERT. My favorite one is biology, but situation, okay. It's okay. Then probably you should be grateful for the situation because it might be leading you to something good. Anyhow. Chalo, my bacha party, 26th, can you guess? It's a direct question from your NCERT, which is why there is no explanation needed. So, I'll tell you one small fact. Okay. Hargobind Khurana was a person who was saying that, okay, the codons which are coming together, these codons are the ones that are going to give rise to amino acids. So, he was in the urge to find what all group of codons are going to give rise to what all amino acids. That was his motto. So according to this, you can tell me the answer. 26A, 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 it is actually 26A. So synthesizing RNA molecules with defined combination of bases. So he was having a defined combination of bases, he was keeping it together and seeing what amino acid is it going to make. So if you're pretty sure about this, you are picking each nucleotide and putting over here. Okay, you're going to put it over here. Now whatever amino acid chain will be made, you will be understanding that okay, this was the codon responsible for this amino acid. So that was his whole theory about. So chemical method developed by Khurana was basically synthesizing RNA molecules with defined combination of bases to find what amino acid does it code for. Okay, so Shagufta, Pratima, BA creations, good. Mamta also, Baba, good. Okay, chalo, let's go with the next question. In a structural gene consisting of 300 base pairs, if 4 nucleotides are added and 1 is deleted at the end point, it leads to what? Now this is again, I don't know how to make lit. Yo, the fire has, this is the fire, okay. This is also a question which is like a fire. Great question it is. 27th. In a structural gene coding for consisting of 300 base pairs. Okay. If 4 nucleotide I am adding and 1 I am deleting at the end point, what will it lead to? Point mutation? Frame shift mutation? There won't be any change. There will be 101 amino acid being made. 27B. What about others? Shweta also. BA creation also is 27B. Chalo, let's solve it. Keep on guessing, okay? I'll tell you the instance. Obviously, I cannot make 300 base pairs over here. I can tell you the instance. Suppose it was AUG and then GAC and then AGC, something like this, okay? This was the normal frame that we had. So this codon was coding for something, this codon was coding for something, this codon was coding for something. Fine. Now what has happened? I introduce 4 nucleotides up here. Suppose it is A, C, G, and then let's make it U. Okay? Now I have put that over here. Let me put that. So AUG is the same. But now the next frame is what? ACG. Okay, and the next frame will be what? There is U also. So UGA. So you see, there is a shifting in the frame. It was read as GAC, but since I have put some nucleotides over here, there is a shift in the frame. So again, it is also going to code for another amino acid, but another amino acid, not the same amino acid. That is there, right? And at the end also, my dear children, what I am doing? I am deleting one thing. So basically it is UGA, okay, UGA, then CAG. In overall sense, I am shifting my frame completely. So this was coding for amino acid, this is another amino acid, another amino acid, another amino acid. So totally protein itself is changed. This mutation is known as frame shift mutation. B. So who all got it? B. Kudos to you. Okay. 27 BA creation started with B. So Shweta, Mamta, Shagufta, Shagadali, everyone, Baba, everyone, great going. Okay. 27 this B, I am so happy. Chalo. 28th question, again something easy for you. Okay. Human insulin is produced in E. coli cells by rDNA technology. That is what? Recombinant DNA technology. This proves that genetic code is what? So human insulin is human's insulin. It is produced in E. coli using recombinant DNA technology. This proves that the genetic code which is there, it is what? It's totally fine, Pratima Baba. No problem. Ayo. Just give me a second, okay? You keep on trying that. Just give me a second. I did something. Now it's fine. 28C, 28A, Baba. So many answers. A, C, A, C. Okay, listen. I already told you. I am a human being. You are a human being. If the DNA which is there in us, okay, it has what? The codons, my dear children. The mRNA which is there in our body, it has codons, that is 3 nucleotides, 3 nucleotides, 3 nucleotides, and all of them are going to go for amino acid, and this amino acid is going to give me a protein at the end. If the same mRNA is being inserted into E. coli and they are also producing the same protein, that means the codon over here is doing the same work there. So can I say they are showing something that is known as universality? It is universality. Basically, the codons which are there, they are universal. We have the same codons like the bacteria. There might be some differences, but most of them, they are universal. So I'll say this shows that it is universal. A is the correct answer. So 28A, 28A, who all got it? A is correct. B creation, Saket Ali, Pratima, Mamta, Shweta, Shagufta. I saw one more person there who was constantly answering, where are you? So now we just have 2 more questions. So I want every one of you to participate. All the 11 people who are watching this. Okay, every one of you should be participating over here. Strict. Have you seen Jasmine Ma'am being strict? Well, no one has seen. So it's okay. So this is the answer. Chalo. Okay, let's say. Which of the following statement is correct, Baba? It's an easy question. Adenine pairs with thymine through 2 H bonds. Adenine pairs with thymine through 1 H bond. Adenine pairs with thymine through 3 H bonds. Adenine does not pair with thymine at all. 29th. What do you think is the answer? So, adenine is friends with thymine and guanine is friends with cytosine. Adenine and thymine, I'll say guanine and cytosine are the best friends out there. Why? Because they have 3 bonds. They are tightly linked. What about adenine and thymine? They have 2 bonds. So what is the answer for this? This is the answer. So that is an easy question, right? So Shagufta, Shagufta, Shakat Ali, Pratima and Jyotika Manjuna. That's a new person in the life. I see you often commenting under the videos. Thank you, dear. And Shweta, 29A, great. Mamta also 29A. Kudos, my dear children. The answer is correct. Okay, adenine and thymine has 2 hydrogen bonds which keep them bonded. If you're asked about guanine and cytosine, it is 3 bonds. Okay. So G and C, okay, how do you remember this? I'll tell you a trick. So G and C both have a curve, they look almost same. So similar people, more close, okay, they are so close that it started looking similar itself. So A and D are not like that, so they have 2 bonds, okay, do not get confused. Chalo. Last question, with this we are going to complete the life. Experimental material in the study of DNA replication has been. So I told you the DNA replication study was done by who all people? This was also a question. Michelson and Stahl. But what were the organism used for this particular study? Hand is spinning. Experimental material, what was it for the experiment? Escherichia coli, Neurospora crassa, Pneumococcus, Drosophila melanogaster. I wish I could give you a prize, but for now I can pray for all of you. That's the only prize that I can give you as of now. So Shaukat Ali 30D, Shagufta 30A. Okay. Drosophila melanogaster, which experiment was done on that for DNA replication? Halima, yes, I was searching for you. Where did you go? Yes. Shagufta, BA creation, Shweta, Jyotika, every one of you is correct. Shaukat Ali, I think there's a typo, error, okay? Or if it is not, then it's okay. Still it's okay. Why? Because I am going to tell you the answer and now we are going to correct it, okay? So experimental material was Escherichia coli, my dear children. Always remember. For Griffith's experiment, who was the experimental material? Streptococcus pneumoniae. Okay. Now, for the next experiment done by this guy, Hershey and Chase, what was it? Bacteriophage and E. coli. Now, for DNA replication, it was Escherichia coli itself. Okay, it was Escherichia coli. So, do not forget that the answer is 30A. Mamta, Pratima, everyone. So, it is A. Please go through the experiment also. You might be asked to do the experiment. If you have not seen my Brahmastra series, you should be definitely watching. I saw all the questions. Okay, so yesterday I did a preparatory exam video, okay, where we got a question paper, we tried to tally it and analyze whether whatever we taught has come or not. So everything, whatever we have taught has come. So Brahmastra series is really important. Go watch it, okay. It will be really good for you. And with this, we are completing the live session. Do you have anything to tell to me? Any doubts if you have, you can ask me right away. Or if you want to tell anything else, you can also tell. Do let me know if it was bad, good, if there is any scope of improvement, or if you need any other help that we need to do from our end, we are ready to do. Give us ideas, we will do it. Or else we will directly end the live. But one thing that I want to tell you guys is I loved being here today for you guys in the live session. So the previous time there were no much audience because probably you had the exams and all, but today I saw a lot of you coming and telling the answers. That's a great gesture, my dear children, because we are working so for you guys. So Shagufta, today's ma'am session was amazing. Thank you so much, Mamta. Thank you. Be a creation. Bye bye. Bye bye, dear. Nice to see you too. So thank you, ma'am. I enjoyed the session. Thank you so much. Next, which lesson? Shaukat Ali is asking. I think it will be sexual reproduction in flowering plants, which carries 11 marks. Yes, should be that. Okay. Thanks, ma'am. I really like your teaching. Oh, thank you, thank you, thank you. Chalo. So we will end. Okay. So see you on some other day. Definitely do not forget to comment, like, and also subscribe to our little channel. Bye bye, dear children. Good night. Sleep tight and take proper care of yourself. Drink water. Take care. Jyotika is waiting for one level up. It's coming at 8 o'clock today. So bye bye.